Welcome to the next session of Principles of Inheritance and Variation. Today we are going to discuss regarding a concept that is nothing but the explanation to the concept of dominance. So, till now we were discussing in genetic class that one of the trait which express itself in the F1 generation is called as dominant, and the trait which or the allele which doesn't get expressed in the F1 generation but appears in F2 generation is called recessive one. So what this concept of dominance, how we can decide which is dominant, or when we call an allele or the trait as dominant, what exactly the dominance is? Let's understand with certain example. See, we know that every gene that contains an information for expression of a particular trait of a character. For example, the height of the individual. So, for example, my height is 5.78 and you may be having 6 feet height. The other may be having 3.5 or 4 or 5 feet height. See, height is a character here. The tall or the short are the traits of this character, right? Like that, every gene contains the information for the expression of a particular trait, or which is required for expression of a particular trait. So, in diploid organisms, such genes will be present in the form of pairs. So, in diploid organisms, the diploid organisms will have the 2 copies of the each gene, or 2 copies of each gene, and such a pair of copies of each gene is called as alleles. And what is the nature of these alleles? Right? These alleles may be in a pair. They may be identical to each other. They may be identical to each other. Or they may be non-identical. So both possibilities are there. They may be identical. If they are identical means that we call it as a homozygous condition. If they are non-identical, that represents a heterozygous condition. See, in case of non-identical pair of alleles, okay, so in non-identical alleles, the one is different from the other. What may be the reason for that one? In non-identical alleles, one of the allele or gene is different from other. What is the reason? Because it might have undergone mutation. Because it might have undergone mutation. What do you mean by mutation? Might have undergone mutation. That is a sudden change in genetic material. Sudden change in genetic material, that is DNA or the RNA. So, this change in the allele or a difference in that allele from the other is because of as it has undergone mutation. So, what that mutation? Mutation is nothing but sudden change in the genetic material. So, because of that only, one of the allele will be different from the normal one. So, what happens because of this mutation? If one is normal allele and other one is the mutated one. So, if the normal allele codes for synthesis of enzyme, that is coding for synthesis of enzyme which is required for the conversion of substrate. This enzyme is required for, so which is required for conversion of substrate. So here the substrate is indicated as S. Nothing but for the conversion of substrate into the product, an enzyme is required, and that enzyme is synthesized based on the information present in that normal allele. So consider normal allele code for the synthesis of enzyme. That enzyme is required for the conversion of substrate S into a product. But what about the mutated allele? But mutated allele code for the synthesis of what are the possibilities here? Why? Because the information present in the allele for the synthesis of that enzyme has got altered because of mutation. So what are the possibilities of coding for a synthesis of enzyme from that mutated allele means? So the possibilities are as follows. That is, 1, it may code for the synthesis of normal enzyme, means even after the mutation also it may code for the normal enzyme, or less efficient normal enzyme only, but its efficiency is less than the normal enzyme. Or it may code for the synthesis of, second option or the second possibility is mutated allele may code for the synthesis of non-functional enzyme. It code for the synthesis of enzyme, but that enzyme is non-functional. And what is the other possibility? The one more possibility is present. What is that? The mutated allele code for the synthesis of no enzyme at all. Means the mutated allele may not code for the synthesis of any enzyme. These are the three possibilities can be expected from the mutated allele. But what is the function of the normal allele here? Normal allele is coding for the synthesis of a functional enzyme, but the mutated allele code for the normal enzyme with less efficient in function, or it may produce a non-functional enzyme, or it may not produce any enzyme at all. So by this example, which allele can be considered as a dominant and which one can be considered as a recessive? Let's see that. Before that, make a note of these points now. See, let's see the explanation for this one. As there are three cases are possible, the mutated allele may produce normal enzyme but less efficient, or it may not produce a functional enzyme, or it may not produce any enzyme at all. So among these three cases, if you observe, in case 1, where normal enzyme or the less efficient enzyme is produced, so modified allele or the mutated allele is equivalent to unmodified allele. So only when the normal enzyme is produced, it produces the same phenotype or the trait as that of the unmodified allele. When the mutated or the modified allele produces the normal enzyme that code for the expression of a original phenotype as that of the unmodified allele. So there is no difference in the modified allele and then the unmodified allele. Is it clear? That is one possibility. That's what happened in the first case. But if you look at the second and third case, in case 2 and 3, what happens here? The allele produces non-functional enzyme or no enzyme at all. So here the modified allele produces non-functional enzyme or no enzyme at all. So because of the non-functional enzyme production or no enzyme formation, here the phenotype may be affected because enzyme is not at all produced, or if enzyme is produced, it is no more functional or it is not a functional enzyme. Because of that, phenotype may get affected, right? So by seeing this, we can say that one unmodified allele which represents the original phenotype. The unmodified allele which represents the original phenotype is called as the dominant allele or the wild type. The allele which produces normal enzyme and code for the synthesis of the normal enzyme and express normal phenotype or the original phenotype, such unmodified allele is called as dominant allele or the wild type. As the same thing happened in modified allele also, this is possible in silent mutation. This is possible in case of silent mutations. Here the allele is mutated, but still it produces the same phenotype as that of the unmodified allele. That occurs during silent mutations. So, if unmodified allele produces the original phenotype, then that allele is called as dominant allele or the wild type. Then, modified allele, which is generally recessive allele or the mutant allele, so only its phenotype may get affected. When the phenotype get affected, the allele which is responsible for that one is modified allele or the mutated allele. So the modified allele generally is the recessive allele, that is called as recessive allele or the mutant type. Is it clear, guys? No. So based on these three possibilities from the mutated allele, so which one we can call as dominant means the allele which code for the original phenotype is dominant allele. The allele which code for the expression of a particular trait, but if the phenotype is different from the original parental type, then that may be the recessive allele or the mutant type. Let us understand this concept by taking the example of the tallness in pea plant. Tallness in pea plant. Before that, make a note of these points now. So, let's take the example as plant height or the tallness in a plant as an example to understand the concept of dominant or the dominance. See, consider the plant height depends on the amount of a particular hormone produced. So, which is responsible for the plant height? The height of the plant will be depending on the amount of particular hormone. So here the particular hormone, if it is produced in maximum quantity, the plant will be having a more height or the tallness. If the amount of hormone synthesized less means the plant will be having a short height or the dwarfness. So amount of hormone production, it further depends on efficiency of process of making it. Efficiency of process of making it means amount of hormone decides the height of the plant, and amount of particular hormone will be depending on the efficiency of process of making it. Let's understand it by taking a live example or day-to-day life example. Say for example, in an pot, okay, some amount of curd is given for you. So what is given for you? Curd is given for you. And from that curd, you have to make the production of butter. So what you have to produce from that butter? If the curd is left as it is in that pot, will the butter will be produced on its own? Andre ಆ ಮೊಸರಿಂದ ಬೆಣ್ಣೆ ತಾನಾಗಿನೇ ಬರುತ್ತಾ? No, right? So here the butter is nothing but amount of hormone. ಇದನ್ನ ಹಾರ್ಮೋನ್ ಅನ್ಕೊಳ್ಳಿ. butter is representing the hormone, right? Or instead of that one, here the butter is nothing but butter is nothing but the hormone. But for the production of that hormone, what is required? The process, that is the efficiency of the process. Means what you have to do? ಆ ಒಂದು ಕರ್ಡನ್ನು ಆ ಕಡಗೋಲು ಅಂತ ಬರ್ತೌದಾ, ಸೊ ಅದರಿಂದ ಕಡಿಬೇಕು. So when you make out of the buttermilk from that curd, then gradually how you go on processing, like that more and more butter will be produced. ನೀನು ಜಾಸ್ತಿ ಅದನ್ನು ಪ್ರೋಸೆಸ್ ಮಾಡ್ದಂಗೆಲ್ಲ ಏನಾಗುತ್ತೆ, ಮ್ಯಾಕ್ಸಿಮಮ್ ಬಟರ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ನೀನು ಪ್ರೋಸೆಸ್ ಮಾಡಿಲ್ಲ, ಬಟರ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದೇ ಇಲ್ಲ, ರೈಟ್? ನೀನು ಸ್ವಲ್ಪ ಪ್ರೋಸೆಸಿಂಗ್ ಮಾಡಿದೆ, ಸ್ವಲ್ಪ ಬಟರ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ಜಾಸ್ತಿ ಪ್ರೋಸೆಸ್ ಮಾಡಿದೆಯಾ, ಎಷ್ಟು ಬಟರ್ ಮ್ಯಾಕ್ಸಿಮಮ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬೇಕಿತ್ತೋ ಅದಿಷ್ಟು ಕೂಡ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ಮೀನ್ಸ್ ಹಿಯರ್ ದ ಅಮೌಂಟ್ ಆಫ್ ಬಟರ್ ಡಿಪೆಂಡ್ಸ್ ಆನ್ ಎಫಿಷಿಯನ್ಸಿ ಆಫ್ ಪ್ರೋಸೆಸ್ ಆಫ್ ಮೇಕಿಂಗ್ ಇಟ್, ರೈಟ್? ಎಷ್ಟು ಎಫಿಷಿಯೆಂಟಾಗಿ ನೀನು ಅದನ್ನು ಆ ಒಂದು ಮಜ್ಜಿಗೆ ಕಡಿಯೋದು ಅಂತ ಹೇಳ್ತಾರೆ, ಅದರ ಮೇಲೆ ಆ ಎಷ್ಟೊಂದು ಎಫಿಷಿಯನ್ಸಿಯಿಂದ ನೀವು ವರ್ಕ್ ಮಾಡಿರ್ತೀರ, ಆ ಮಜ್ಜಿಗೆನ ಕಡಿದಿರ್ತೀರೋ ಅಷ್ಟು ಬಟರ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ಇಫ್ ಯುವರ್ ಎಫಿಷಿಯನ್ಸಿ ಈಸ್ ಲೆಸ್, then less amount of butter will be produced. Like that, don't write that as an example in your examinations. Is it clear? This is just to make you understand this concept. Now let's consider. So amount of production of particular hormone depends on efficiency of a process of making it. Then amount of hormone decides the plant height. Then now consider the enzyme is responsible for this process. ಹಾಗಾದರೆ ಈ ಪ್ರೋಸೆಸ್ಗೆ ರೆಸ್ಪಾನ್ಸಿಬಲ್ ಆಗಿರ್ತಕ್ಕಂಥದ್ದು ಯಾವುದು? ಎಂಜೈಮ್. ಇಫ್ ಎಂಜೈಮ್ ಈಸ್ ರೆಸ್ಪಾನ್ಸಿಬಲ್ ಫಾರ್ ದಟ್ ಪ್ರೋಸೆಸ್ ಆಫ್ ಮೇಕಿಂಗ್ ಆಫ್ ಎನ್ ಹಾರ್ಮೋನ್, ದೆನ್ ವಾಟ್ ಆರ್ ದ ಪಾಸಿಬಿಲಿಟೀಸ್? If this enzyme works efficiently, means the gene coding for that particular enzyme works, is normal or the unmodified allele, and produces the enzyme which can work efficiently. In one case, when the allele or the gene which code for the synthesis of the enzyme is normal, so the enzyme will work efficiently. When that enzyme work efficiently, a lot of hormone will be produced. A lot of hormone will be produced. When the efficiency of that enzyme is more, so it produces more, or it make out more hormone production. When the plant hormone is produced in more quantity, then so the plant will be, so such plant will be tall in nature. Why? Because the hormone which is responsible for that height of the plant is more, so the plant will be tall. This is possible when the enzyme which is responsible for the make out of that hormone is very efficient. Let's see the other possibilities. If the gene which code for the synthesis of that enzyme is altered or mutated, then what happens? It may code for the synthesis of enzyme which is non-functional, or it may not code for the synthesis of any enzyme at all. So mutated gene code for the synthesis of enzyme which is non-functional, or the mutated gene may not code for the synthesis of any enzyme at all. ಎಂಜೈಮ್ ಸಿಂಥಸೈಸ್ ಆಗಿದೆ ಬಟ್ ಫಂಕ್ಷನಲ್ ಇಲ್ಲ, ಅಥವಾ ಯಾವುದೇ ಎಂಜೈಮ್ ಸಿಂಥಸೈಸ್ ಆಗದೇ ಇರ್ಬೋದು. ವೈ? ಬಿಕಾಸ್ ಜೀನ್ ಹ್ಯಾಸ್ ಗಾಟ್ ಮ್ಯೂಟೇಟೆಡ್ ಆರ್ ದ allele is mutated or altered. Then what happens? Because of this, when the process is not efficient, so what happens? No hormone synthesized in less quantity. So hormone synthesized is in less quantity, or no hormone is synthesized. No hormone synthesized. These are the two possibilities are possible. ಯಾವುದೇ ಹಾರ್ಮೋನ್ ಸಿಂಥಸೈಸ್ ಆಗದೆ ಇರ್ಬೋದು, ಅಥವಾ ಸಿಂಥಸೈಸ್ ಆದಾಗ because of its less efficiency, less amount of hormone may be synthesized. So as the result of that one, what happens? So the plant will be short or dwarf. So that what happens? Such plants will be short or the dwarf. So here which example is considered? Plant height. Okay, and we know that the tallness is an dominant character or the dominant trait of the character, and dwarfness is an recessive trait or the recessive trait of the particular character called as plant height. So which is dominant and which is recessive here? The gene which code for the enzyme which work very efficiently and that is responsible for the tallness of the plant, such gene is called as dominant. And the gene which has got altered or mutated, so it code for the non-functional enzyme or no enzyme at all. As a result of that one, the plant is found to be the dwarf one, and such gene is called as recessive. So this is how the dominance and then the recessiveness of a particular allele can be predicted or considered. Is it clear, guys? So this is regarding the concept of dominance by taking an example. So here what we got to know means unmodified allele is called as dominant allele, which represents or which produces original phenotype as that of their parents. And if the allele is modified and code for the synthesis or the production of affected phenotype, then it is called as a recessive allele. Clear, guys? So this is what for today's class, and in next class we are going to discuss regarding the two more concepts which represents a deviation from the Mendelian principles, that is codominance and then the multiple allelism. Thank you, guys.